The role of serotonin and histamine in an animal model of depression. Might it take two to tango? Chronic stress is an important public health concern, especially given the growing number of stress-induced mental health disorders such as depression. Traditional treatments such as selective serotonin reuptake inhibitors (SSRIs) have not been effective for everyone, leaving a gap in the treatment of many people living with depression. On one hand, researchers wondered whether depression is caused by chemical dysfunction in the levels of brain messengers called neurotransmitters that are responsible for mood, especially serotonin. They call this theory monoamine hypothesis based on the fact that serotonin is a monoamine. On the other hand, more recent research suggests that inflammation, process triggered by the immune system in response to things like illness, but also stress and the release of linked bodily messengers that help with inflammation, pro-inflammatory cytokines caused by chronic stress, may have an impact on serotonin levels. Similarly, researchers found that histamine, which is a chemical in the body often associated with allergies and immune activation, can be influenced by serotonin levels linked to stress-induced inflammation. The group of researchers around Melissa Hersey and Melissa Renew from the University of South Carolina and Imperial College London have published their work in 2022, and their findings were intriguing. So what did they do? The researchers used a chronic mild stress (CMS) method to induce stress in male and female mice over a 16-week period. This model was used to stimulate the unpredictable and mild stressors that could lead to depression-like behavior. Behavioral tests were done to check anxiety and depression-like behaviors. First, researchers measured the levels of pleasure of mice using a performance test to measure so-called anhedonia, which is a lack of pleasure indicating stress and depression, via the sucrose sugar preference test (SPT) using sugary drink. Normal and happy mice will prefer this over water, but unhappy mice won't show a particular preference. Then, they used behavioral tests including the elevated zero-maze EZM to induce and measure anxiety. EZM is a test where there is a cycling corridor elevated above the floor with 2 open areas and 2 closed ones. Mice don't like open and elevated areas, but they are also curious. Researchers can estimate how anxious or relaxed a mouse is by measuring how long they hang out in closed versus open areas. The third test is the forced swim test, FST. The FST is a test where a mouse is placed in water. Mice don't enjoy that very much and are fearful of drowning, so will usually frantically try to swim and escape. However, an unhappy mouse will stop struggling more quickly. Fourth, the tail suspension test, TST, to assess depressive-like behaviors. The TST kind of does the same thing, but instead of being put into water, the mouse is dangled by their tail, which they ordinarily would try to fight. The researchers measured serotonin and histamine using advanced voltammetric technologies, which are very clever as they make it possible to measure brain messengers or neurotransmitters levels in real time. These measurements were made in the hippocampus and hypothalamus, areas of the brain which have been fairly consistently been linked to stress response and mood regulation. The study also employed machine learning to tell apart stressed and non-stressed animals based on how much serotonin they had in their system. And a computational model was developed to predict how inflammation of the brain triggered by stress affects the serotonin and histamine. An easy example of computational model is weather prediction. The model might simulate weather patterns by taking into account factors like temperature, pressure, humidity, and wind speed to predict upcoming weather. So, in the case of this study, they wanted to be able to guess correctly or predict which animals had been stressed by how much serotonin they had. So, what did they find and what happened next? The researchers found that all mice subjected to chronic stress showed lower levels of serotonin in the hippocampus, regardless of how they behave in the tests. This was an important finding, as it suggested that serotonin could serve as a good biological marker of chronic stress. They also found that the more stressed mice became, the more they showed depression-like behavior in the tests. On the opposite, stressed mice showed higher histamine levels. The researchers then concluded that brain inflammation caused by stress increases histamine activity, which in turn limits serotonin release. One goes up, the other one down. These findings led the researchers to develop a clever measure, the stress index SI, a new completely mathematical model of stress, which is based on what they had just found out about the relationship between serotonin and histamine levels, suggesting that decreasing histamine while increasing serotonin would be the most effective way to restore serotonin levels to normal in stressed mice. They did not stop there. They actually tested the idea to see if the model's predictions were accurate. Mice were treated with citalopram, an antidepressant drug that is supposed to increase serotonin levels, and with MH, a drug that stops histamine from being produced. And guess what? The combination treatment was able to restore serotonin levels to near normal, supporting the model's suggestion that targeting both serotonin and histamine brain pathways could be a more effective treatment for stress-induced depression. What a finding! This research highlights the potential for serotonin and histamine to serve as biomarkers of chronic stress and stress-related mental health disorders. Often, behavior or symptoms reports are the only way of determining the stress, so adding in the biological perspective and being able to measure objectively are a very exciting prospect. This is because they won't just enable us to diagnose potentially, but also to monitor treatment success and identify health risks, which there are many of in context of chronic stress. And that's not all. By understanding better the interplay between messengers and inflammation in the brain, this study suggests new ways in which we can find more precise and effective treatments for depression, particularly for individuals who do not respond to current serotonin targeting therapies. And there is quite a few of those people. And this brings us back to where we began, serotonin and histamine. Might it take two to tango?